Hello, this is Sana Arshad, a medical laboratory scientist, and today I'm going to deliver a lecture related to culture media that are used routinely in a diagnostic microbiology laboratory. I'm trying to teach in an easiest way of which you can understand clearly and distinctly. And at the end of this lecture, this distinct concept will let all students, as well as people who are working and practicing in diagnostic medical laboratory. So let's get started. So first of all, we have to understand what is the culture media. Actually, it's a special medium in which there are essential nutritional requirements that aid the growth of microorganisms. And these microbes use these nutrients as their food for rapid growth, as well as their number is also increased. So there is another term which is agar. This culture media, which I just described, is composed of jelly-like substance, and this jelly-like substance is known as agar. So different nutrients and chemicals are added to this gel-like substance to aid the growth of microorganisms. So these nutrients can be blood, serum, yeast extract, peptones, and certain growth factors are added, etc. etc. There are several ways of categorizing culture media, but right now I'm going to discuss those culture media that are routinely used in a microbiology laboratory. Before going ahead, I would like to talk about the picture that is on the right side. This picture on the right side shows cooked meat media, which is an example of enriched media. And I will explain enriched media later. This media supports the growth of both fastidious and non-fastidious microorganisms. In diagnostic labs, the remaining sterile specimens after processing are transferred to CMB. I would like to explain this with the help of an example. Let's say CSF specimen comes to a lab and CSF is cultured onto blood, chocolate, and meconic agar. And if these media will not yield any growth, then the remaining specimens that had been cultivated in CMB will aid to isolate the pathogens by subculturing from this enriched media, which is known as CMB, cooked meat media. The first one is basic media, and the basic media is mainly used for microorganisms that don't require essential nutritional requirement to grow. What's the purpose of basic media? Actually, this media is used to stock cultures for control strains. The control strains may be of enterobacterials, staphylococci. The second main use of basic media is that it is used as base for enriched media. So all the fastidious microorganisms cannot grow on this medium because they require extra nutrients to grow. So the examples of basic media are nutrient broth, peptone water, and nutrient agar is also one of the example of basic media. Nutrient agar is a basic medium for growing non-fastidious microorganisms, and nutrient broth, it is actually a liquid medium. And the difference between nutrient broth and nutrient agar is that the concentration of agar in liquid medium, that is nutrient broth, is less than that of nutrient agar. So this is only the main difference of broth and nutrient agar. So nutrient agar, it is actually a base of blood agar and chocolate agar. The picture on the right is of nutrient agar. Now I'll ask a question from you. Can you tell me by visualizing the growth pattern on this medium? This is a typical swarming growth pattern of Proteus mirabilis. So now we will talk about enriched media. Enriched media contains the nutrients that aid the growth of wide variety of organisms, including some of the more fastidious ones. So in basic media, we add blood, serum, certain growth factors, yeast extract, vitamins to make it enriched media. So the fastidious organisms are Streptococcus species, Haemophilus influenza, Neisseria species, and they require special nutritional requirement. So what are the examples of enriched media? Blood agar is an enriched medium in which whole blood supplies all the nutrients to aid the growth of these fastidious ones. And chocolate agar is also an enriched medium. And why it's called chocolate? It's because in chocolate agar, heat treated blood is added and it turns to brown. The color of the medium turns to brown. That's why it's named chocolate. The next one is enrichment medium, and it allows only target organisms to grow while inhibit the growth of unwanted microorganisms. And this inhibition of unwanted organisms is temporary, so we have to subculture it within specific time frame. So example is selenite F broth that is used for salmonella species, and within 8 to 10 hours we have to subculture. Otherwise, after 8 to 10 hours, commensals will start to grow. So here you can see 4 tubes. These are enrichment media. The first one is selenite F broth that is used for stool specimens. And the second tube with orangish color, this is selenite F broth after overnight incubation. And the third one is Todd-Hewitt broth, and this is enrichment medium for group B streptococci. And the fourth one is also Todd-Hewitt broth, but it is after overnight incubation. And Todd-Hewitt broth is also one of the example of enrichment medium. Next comes selective medium. As the name indicates, it allows the growth of only selected microorganisms. So these media favor the growth of desired microorganisms and inhibit the growth of undesired organisms. This medium can be made selective by adding antimicrobials, pH, or chemicals. Let's consider example of antimicrobials. In chocolate agar, if we add vancomycin, colistin, and nystatin, then it becomes selective for Neisseria species. Similarly, another example is that if we add in sheep blood agar polymyxin B and nalidixic acid, it becomes selective for gram-positive microorganisms. And the pH of seborrheic dextrose agar is maintained at 5.6 to yield the growth of yeast while it inhibits the growth of bacteria. The pH of seborrheic dextrose agar is maintained at 5.6 and it helps to grow yeast while it inhibits the bacterial growth. Let's consider an example of chemical added to a selective media. Serum tellurite agar that is used to grow Corynebacteria, while the tellurite added to it inhibits the growth of other microorganisms. And here are the pictures of selective media. The left side picture is of XLD and the right side is of McConkey, and the lower one is XLD with growth, and the colonies are of lactose fermenting E. coli. Next comes differential media. It is also called indicator media. As name indicates, it is used to distinguish between microorganisms. And the indicator is added to the medium, such as neutral red, phenol red, methylene blue. That indicates the defining characteristics of a microorganisms. And in this way, specific organisms are promoted to grow by changing color of the media with change in pH of the color of the medium. And here are some pictures of differential media, and the top one is XLD, and you can see the growth of salmonella. The black color colonies are of salmonella, and the black color is due to production of H2S. The second one is biplate, and you can see two mediums on one plate. The left side is blood agar and the right one is clad agar. And here urine specimen is inoculated on biplate, and you can see the growth of E. coli. And the third one is McConkey agar, and the magenta color colonies on McConkey agar are lactose fermenting colonies, and the other one are non-lactose fermenting colonies. The next one is transport medium. Transport medium is used to keep microorganisms viable if there is delay in specimens processing. So one of the example of transport medium is Amy's transport medium, if amines transport medium is without charcoal, it maintains bacterial viability for 24 to 48 hours, and the amines transport medium with charcoal keeps the fastidious bacteria such as Neisseria haemophilus viable for 24 hours. So another example is carrier medium, and it is used for enteric specimens. And the third one is alkaline peptone water, that is used for stool specimens and is specific for Vibrio cholerae. The next one is identification media. This media is basically used to identify bacteria, and it contains indicators and substrates. The color of the medium is changed because of indicator added to the medium. Examples of identification media are chrome agars. The chrome agars are of different types. One of the type is MRSA strains, that is methicillin resistant Staph aureus, and in MRSA, that is methicillin resistant Staph aureus, there is oxacillin added, and the resistant strains of Staph aureus will grow on this medium, while MSSA will not grow on this medium, that is methicillin sensitive Staph aureus, that will not grow on this medium. The second chrome agar is vancomycin resistant Enterococcus, and there are chrome agars for UTI pathogens. Similarly, there are chrome agars for Candida. And identification media also include biochemical media such as TSI, urea agar, citrate agar, etc. Let's understand identification media with the help of examples that are shown on the right in the form of pictures. The first one is API, and this API is specific for Neisseria species. The second one is TSI, and you can see three biochemical reactions. The first one has yellow slant and yellow bud, and it indicates the growth of lactose fermenting bacteria. The second one has red slant and yellow bud with H2S production, and it indicates the growth of salmonella. And the third one, there is no change in color, and these are non-fermenting bacteria. On the lower left, you can see bile esculin agar. The black color of the medium indicates the positive reaction, and it is enterococcal that hydrolyzes esculin. That's why the color of the medium changes to black. And the other one indicates negative reaction. The lower right picture is of indole reaction, and the tube which indicates haziness is motility positive, and you can see the change in the color on the surface, which is pinkish red. So this tube indicates indole motility positive reaction, and in the other tube you can see there is no haziness. So as there is no haziness, it indicates non-motile bacterial growth, and at the surface after adding indole there is no change in color, and it is indole negative. This tube indicates negative indole motility reaction. I hope so at the end of this lecture you will understand this topic in a much better way. I'm always there for you. If you have any question, you are free to ask by typing in the comment box. Have a great time.